Hello there, how are you doing? It's my wonderful pleasure to have you guys with me today on the lecture. So I'll be solving compilation of questions on paper one. So this video will be in two parts, I think. I may make it three parts depending on how long it is it took me to solve the questions, because I'm going to explain some things a bit further, because this is towards those writing GCSE, so it's meant to be a bit thorough. It's advisable you've seen my videos on each of these topics, or you've read it by yourself, but it's still okay. I'll still try to make you understand some things, but preferably it's always better if you've actually read the topic. And then this is how it's done. If you are seeing this for the first time, you should first attempt the question by yourself, then afterwards wait and see what answer the question, then see what I'm going to say, or just pause the video to do that. All right, this question is about cells. Okay, figure one, which shows a cell. So it says, what type of cell is shown in figure one? Talking about this very cell here. So, well, apparently it is a bacterium cell. Why? Number one reason is because the DNA loop is not in a nucleus. So, if you can remember, organisms that doesn't have nucleus are called prokaryotes. All right, while the ones that have nucleus are called eukaryotes. Another thing we can see is that it has this should be whether this or this, that should be the circular DNA called plasmid. I think it's this one. That should be circular DNA called plasmid, which is used to develop resistance against antibiotics. It's a common question. Develop resistance against, excuse me, resistance against antibiotics. So basically that's what it does, antibiotics. So that's the thing. The another thing it has cell wall. So especially this and these are peculiar to bacterial cells. So, and that's why we picked that. We'll have picked plants if we're looking at this, because plants also have cell wall, but plant doesn't have its DNA in the cytoplasm, because this one is just in cytoplasm. It's not protected, as it were. So the next question says, what is the function of the cell wall? You're meant to tick one. So of course, this is the cell wall in the cell. So, well, the function of the cell wall is to strengthen the cell. All right, then that's basically other things to contain the genetic material. That would be the nucleus and all of that. So let's move on. Which part of the algal makes it green? Talking about this, because this question continues as this. So what part of this makes it green? Well, whether you even see the picture or not, we know that what makes the plants or any of those things appear green will be the chloroplast. All right, that made sense. Moving on. Now, the student looked at another cell, and the image width of the cell was 40 millimeters. So this is the image size. Then it says the real width of the cell is 0.1 millimeter. Okay, that's the actual size, as it were. If you can remember this triangle, it's always good you remember that. It's just a quick reminder. It's always you have MIA, magnification, then we have image over actual size. So for you to get your magnification, is going to be magnification equals to image over actual size. For you to get your image, is going to be image equals to magnification multiplied by actual size. For you to get your actual size, it's going to be image over magnification. So it's just like once you cover if you cover this, then you have this. If you cover this and all of that, so basically it's just the aim is for you to see the whole thing, as it were. It's just a quick reminder. So let's put this here. So from what we have here, now let me say it here. This is quite very straightforward. This was okay. This is both straightforward because it's just going to be multiplication. We just say that would be okay, image size, that's going to be 40 millimeter over 0.1 millimeter. So the millimeter cancels each other out. This in this is going to be 400. So it's going to be times 400. But if we're going to make this more difficult, like I did to my students in class, I turned this into micrometer. You might, I want you to just pause the video right now and try doing this question when it is micrometer. Now, so if it is micrometer now, so that means our image size is 40 millimeter, then real size or actual size is now 0.1 micrometer. And we should remember that 1000 micrometer makes 1 millimeter. For that reason, when you are doing a question like this, the two units must be the same. So we have to convert this into millimeter. So if we do that, that will give us 0.0001 millimeter. So if you put the value, so it's going to be 40 millimeter over 0.0001 millimeter, as it were. Let me write that clearly. So if you do that, if you divide that all the way, it means that you're going to have so millimeter cancel is going to be 4 times 400,000 times. Yes. So that's what's going to be. So the idea here is that always know that you're supposed to have the unit should be the same. It must be the same. It has to be the same. If you convert this into nanometer, you're still good. So I mean to micrometer is still good. Just that you're just going to have to multiply it, so to say. So it's fine. Mind you, maybe you're wondering, how did I get this? What I did was that it was 0.1 divided by 1000. That's how I got this here. But if we're going to convert this to micrometer so that it will be the same with this, there's going to be 40 times 1000. That's going to be 40,000. So you still get the same answer as it were. So it's just it's fine. But usually just make sure that the units are the same. Moving on. Now it says here, draw a line from each stage of the cell division of the cell cycle to what happened during the stage. So these are the stages here. These ones are the stages and these are the events. Bear in mind that this is not all the stages, just an extract from it. So stage 1, that will be this one. Yes, the cell grows and chromosome replicates. Yes, that's what happens. Then in stage 2, one set of chromosome is pulled to the other side. Actually, this is called anaphase. This particular thing is called anaphase. Then this one is called interphase. This is under a phase called interphase, a stage called interphase. Then the last thing that happened is the cytoplasm and the cell divides, and that process is called cytokinesis. Cytokinesis. All right. So let me give a quick overview. In cell cycle or cell division, whether it is mitosis or meiosis, the cell will usually go through something. So cell, let me just write cell cycle is usually divided into two, which is going to be interphase and PMAT. Sorry, is and mitosis. Mitosis. And that mitosis is divided into PMAT. The P M-A-T. The P is prophase. The M is metaphase. Sorry, I didn't write that very well. The A is anaphase. And the T is telophase. Now, after this telophase, then we have this separate process called cytokinesis. This cytokinesis sometimes cytokinesis. Sorry, I didn't let me write that clearly. That's not clear enough. Okay, this is the same thing here anyways. cytokinesis. The point is, some book will add this cytokinesis as like the last stage of telophase. Well, it depends really, but it's something that should be, it's a bit different in a way, yes. So now you might want to check up my video on cell division. It's going to really help you here because I can't go into details. This is a topic in itself as it were, all right? So, but just check out my video. I think by this time I should have uploaded. If I have not, just watch out for it, all right? So basically, this is the different stages as it were. Now, let me say something real quick here. This is the same thing you still have in meiosis, but it's going to be a bit different. Like I said, just check out the video. It's going to make sense. This question is about cell division. Which process makes two identical new cells for growth and repair? Well, growth for two identical cells, or even growth and repair, either of these two, if you only have this, only this, even without this, you still good to pick mitosis. And if what you only had was this, you're still good to pick mitosis, because mitosis is the only cell division that is what we use to grow in size and also to repair damaged cells and all of that as it were. So the next thing here is it says a cell divides to form two new cells every 24 hours. How many days will it take for the original cell to divide into 8 cells? Okay, if you're going to do that, don't forget one cell forms 2 cells in 2 hours, in 24 hours. How many days will we use to get 8 cells? Well, the answer is going to be 3 days. How? A cell, this is a cell, is split into 2 in 24 hours. But don't forget that each of these cells will split again. So into 2 in 24 hours, each of them. 2 like that. So in another 24 hours again, we then have this one splitting as well into 2. Each of them have to split into 2. All right. Then so this is day 1, day 2, day 3. So at the end of day 3, we have 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. The reason is because the cell kept one cell, we have to split into 2 again. So it's not as though it's just one cell dividing. I mean, once it divides, we have 2 coming out from that, because this divided again and this will divide by itself. That's why we have that. That's why I have to draw this whole thing. So it's going to take 3 days for you to have that, as it were. Now it says here the chromosome contains genetic material. Name the chemical which the genetic material is made from. That is deoxyribonucleic acid, for short, DNA. If you write DNA for short, I think it's okay. I just feel like writing that in full. Now, the genetic material is made from small sections. Each section codes for a specific protein. What is one section of this genetic material on a chromosome called? That is called a gene. Let me say something real quick here. Always remember this hierarchy. Nucleus contains chromosomes, or we can say chromatin. It's the same, but chromatin is just like the thread form or uncoiled, or let me say uncondensed form of chromosomes, as it were. So inside chromosome, or let's say within chromosomes, you have DNA, which looks somewhat like this, or sometimes we draw it like this. It has no nice. I'm coming. Like we just draw it like that. So it has this double helix. I didn't draw that very well anyways. So now I'm going somewhere. Within this DNA, we have sections called genes. So we have genes. What does the gene do? The gene is what helps us to code for protein. What's the essence of protein? Protein is responsible for our traits and characteristics as well. All right. When I mean trait character, I'm talking about everything you do, the shape of your eyes, your finger, your behavior. Yeah, that's what happens. That's what trait means. All those features, whether it is inside or outside, or the things you can see, and all of that. Okay, the next question says stem cells are cells which have not yet specialized to carry out a particular job. Bone marrow cells are one example of stem cells. Okay, explain how a transplant of bone marrow can help to treat a medical condition. Well, let me say here to start with, bone marrow is an adult stem cell, so it is partially committed already. We have something called embryonic stem cells. This embryonic stem cell can form any type. We call it unspecialized. It can form anything. It can form bone cells. It can form muscle cells. It can form uh, anything. Brain cells. It can form anything. But bone marrow stem cells can only form types of blood cells. It can form red blood cells, it can form white blood cells, it can form platelets, but it can't form say bone cells. So that's why I said here that you should remember this is an adult stem cell. So your answer cannot be something here. You can't say to treat a wide range of diseases. So let me give you the answer right now. So it's going to be this can be used to treat blood diseases. And the reason is because it can only form a type of blood cells because it's partially committed already. This can be used to treat blood diseases such as sickle cell anemia. So this is this can form any type of blood cells. Don't forget, you can't say it can form any type of cell, because if you say that, you're wrong, because bone marrow cells are not pluripotent. Pluripotent means they can't form everything. They are partially committed. They can form different type of blood cells. They are not just red blood cells only, but they can form any type, but it's just within the blood cells, not something very wide as it were. Moving on. There are two types of cell divisions, mitosis and meiosis. Describe differences between the processes of mitosis and meiosis. 3 marker. Okay, let's start with mitosis. Mitosis yields two daughter cells, while meiosis yields four daughter cells. All right, that's one mark. We can say again, mitosis is for growth and repairs, while meiosis is for sexual reproduction. Yeah, to form gamete as it were. Then we can say that mitosis has one stage, while meiosis goes through two stages. Then I can go on and on, but let me just add one more, which is you can say that mitosis produces identical cells, while meiosis produces cells with variation. This is very, very important. So now let me say, I want to explain this real quick. That meiosis, so mitosis is just, so when you say mitosis, it's just this stage, PMAT, which is prophase, metaphase, anaphase, telophase. But if it is meiosis, it's going to be, if it is meiosis is going to be two phases. Meiosis 1, which is divided into P, PMAT 1, then meiosis 2, which we then have PMAT 2. Usually we don't write that way, we write it this way. So it's going to be, so it's the same prophase. So it's going to be under meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. Under meiosis 2, you have prophase 2, metaphase 2, anaphase 2, telophase 2. Basically, that's just what that means there. What is meant by the term diffusion? Always please take note of the answer, the mark. So it says here, that's two markers, so you're not supposed to just define. So this is the movement of substances or molecules from area of high concentration, let's say higher concentration, to an area of lower concentration, period. Now, that's one mark. Second mark is it does not require energy. Third point, this is a bonus, it is used to transport substances like glucose, carbon dioxide, oxygen, then electrolyte, so to say. Okay, electrolytes, e.g. sodium, potassium, calcium, and all of that. Those are the things that diffusion can be used to transport in and out of the cell, because diffusion is a type of cell transport, or transport across cell membrane, as it were. So this will be the end of part 1.